Hello class, today we're doing lecture 8.4 and that's going to be on RNA and protein synthesis. In this lecture we should be able to tell how RNA differs from DNA. We should be able to name 3 types of RNA. We should be able to describe transcription and editing of RNA, and we should identify the genetic code. So the differences between RNA and DNA. We'll start there. mRNA is a disposable copy of a DNA segment, so it's only part of a DNA segment. RNA is similar to DNA, except mRNA is 1 strand and DNA is 2 strands. So we have RNA here, DNA here. Also, RNA has uracil instead of thymine. So we have uracil instead of thymine. And the base pairs go adenine and uracil and guanine and cytosine. So it's just changing the T to a U. Now how I always remember that is just the alphabet. It's moving 1 letter down. Also another similarity, okay, is that RNA has ribose instead of deoxyribose. So here's their chemical structure. And we have deoxyribose here. And we have just ribose here. And the main difference is highlighted right here. And that's because this is an alcohol group and this one's just a hydrogen group off the second carbon. And how I know it's the second carbon is because it's labeled, and this is all chemistry stuff. So RNA is ribose, so just remember R, and DNA is deoxyribose, and it's D and D. RNA is going to control the assembly of amino acids into proteins. So we all know that amino acids make up proteins. That was way back in chapter 2. So RNA actually controls how those amino acids are linked together to make the proteins. There are 3 types of RNA, and they each have a specific job. First off, messenger RNA, also called mRNA. And that is going to carry information to create proteins. Okay, and messenger always starts at the 5 prime and it starts to code itself, which it just did, and that would be the messenger RNA. So here it goes again. It starts to code itself from the 3 to the 5 prime. And it codes with whatever it needs to be based with. Okay, you see right here where the U came to the A instead of a T? That's where the uracil is. So we know that's messenger RNA. That's an RNA if there's a U in it. If there's a T in the DNA, it still messages with an A, combines with an A. So this is a great little visual for you to look at if you need help with looking at mRNA. The next one is going to be ribosomal RNA, also called rRNA, and that makes up major parts of the ribosomes. And the ribosomes we know is an organelle that is responsible for protein synthesis, so the breaking down of proteins. So here are ribosomes going in and then breaking off. The next one is tRNA, which is transfer RNA, and that transfers amino acids to the ribosome. So here we have the ribosome, and we have the tRNA coming in to transfer that amino acid. So let's talk about transcription a little bit. Transcription is the biological process of copying a portion of the original DNA molecule, and it produces mRNA. So you need to know that transcription is going to make mRNA, or messenger RNA. It occurs in the nucleus, and a sequence of DNA is copied into a complementary sequence into mRNA. It requires the enzyme RNA polymerase, which we saw in the whole little video there. So during transcription, promoters are signaled in DNA that indicate to the enzyme where to bind with the RNA. Introns, which are DNA of a eukaryotic gene, contain sequences of nucleotides that are not involved in coding for proteins. Exons are the DNA sequence that code for proteins. So the introns are cut out from the RNA molecules while in the nucleus, and the remaining exons are then spliced back together to form the final RNA. So the roles of DNA and RNA, okay? Cells use the vital DNA, or the master plan, to prepare RNA, which are the blueprints. So the RNA is the blueprint to make the big master DNA. DNA remains in the nucleus, and RNA molecules go to the protein building sites in the cytoplasm, which are ribosomes. Most genes contain nothing more than instructions for assembling proteins. Proteins are the microscopic tools, each designed to build and operate a component of a living cell. The genetic code. Polypeptides, we talked about polypeptides in chapter 2, okay? Polypeptides is a protein made by joining amino acids into a long chain. Genetic code is a language of an mRNA, and it's a code that is read in 3 letters at a time. A codon is a 3-letter groupings of genetic codes that represent different amino acids. So you should know what a codon is, and we're going to learn on how to read those. So in this RNA sequence right here, we're going to break these up into 3-letter codes. So we have UCG, which breaks down, CAC, which breaks down, and GCU, which breaks down. Okay? And each of these is a specific polypeptide or amino acid group. And I'm going to teach you on how to come up with the names. We have a little chart to help us. So here is our little chart. And how to use this genetic code diagram, you start in the center and work out. So first one, GAG. So we're going to start and we're going to find G. So we're going to find G. And then we're going to go to the next, the lighter yellow, and we're going to find A. And then from there, we find the next letter, and it is G as well. So then what it would be glutamic acid. Okay, so for the next one, we find A, then we go to the next letter A, and then we go to the next letter A, which is lysine. Okay, in your book it has the same thing, and it has this circle. And I will give you a little worksheet to practice on using this as well. And that is where we're going to stop for today. If you have any questions, please make sure you write them down in your notes and then ask me them. Have a great day.